Okay, thank you very much for that kind introduction. It's lovely to be here today, especially because, as I've seen already in my lab visits and meetings today, so much really good microfluidics is going on. So I'm going to apologize up front if I go over some ideas and some concepts that many of you already know. But really what I'd like to do today is tell you a little bit about work that we've been doing in my group now for quite a few years, both at Imperial College London and at ETH Zurich, which is really focused on using very, very simple microfluidic systems to do chemistry and to do biology with a very simple view of improving efficiency to some extent. So you'll see examples of chip-based systems, of capillary-based systems, but the real idea or the concept behind a lot of the work that we do is, can we add advantage? So really, to begin, I'd like to remind myself as well as you why we might think about using miniaturization. So downscaling large-scale instruments to the small scale. What are the key advantages? And just to give you some reference here, these are examples, very old examples, in fact, of devices used in our group, which are focused around, in both cases here, rigid materials. This one is in glass. This one here is in silicon and glass. And these are the kinds of systems that we use to do both analytical and reactive processes. So you can see here very simply that we have features such as channels here, mixing regimes in this case here, which allow us to mix and process fluids through cross-sectional dimensions of a few microns. So these are typically very simple systems. In all the examples I think I'm going to show you, apart from one, we use passive microfluidics to affect our operation. So as you'll see, we're simply going to use a control of the hydrodynamics and of the geometry of the system to affect a process. But if we just look at this very short list here of some of the major reasons why we might want to miniaturize, some of them are very obvious. So the first points here in black, the ideas of the cost economies through micromachining, the reduced sample, reagent and power consumption, and portability are very, very obvious. I guess here, in terms of reduced sample and reagent power, this is relatively important for what I'll talk about today. The instantaneous volumes that we work with within our systems are typically no more than a few tens of nanoliters and are more normally on the order of a few tens of picoliters. So these are very, very small. But if we go down to basics, really, the bullet points here in blue that I've written here are the real reasons why we've been using microfluidic systems for a number of years now. So the first one here is superior performance, and by that I mean that typically as we shrink systems down to the microscale, things typically get faster, they typically get more efficient, and also we can exert a degree of control over a process in a way that would be very, very difficult on the large scale. So this is a primary driver for the work that we do, and hopefully we'll see examples of this afternoon. We also have the ideas of facile process integration and automation. Can we daisy chain or can we integrate a number of different processes together? And that then should allow us to do automated experiments where we can really reduce error, so we can improve the robustness and reproducibility of a measurement. And then two aspects which I think are particularly important that I'll highlight today, hopefully. The first one here is high analytical throughput. Can we think about doing as many experiments as we can per unit time? And linked into that, can we think about using microfluidics to do things in a completely different way? So I'll show you again, hopefully, examples of processes that would just be impossible to perform on the large scale. So rather than use that very long-winded list here, perhaps we can think about this simple statement at the bottom. And this really defines the core reason why our group has been interested in using microfluidics. And that's the fact that using very simple microfluidic systems, we can generate very high quality chemical and biological information, and we can do that very, very quickly. So my last introductory slide, and again I apologize for the simplicity, but at least it reminds us of some of the key features of the microscale, is just to think about how our flow environments really differ from the macroscale. So the first thing that we can think about here is the idea of high surface area to volume ratios. This is something that we all know, but what does it mean for the experimentalists? For us, it very, very simply means that we can move heat and we can move mass around very, very efficiently. So as a chemist, we can maybe create or homogenize solute and temperature gradients on timescales that we just couldn't access on the large scale. So that's a first very important reason. But also, as we all know, that as scale decreases, fluids basically are increasingly influenced by viscous forces. So viscous forces dominate inertial forces, and this we would normally characterize by something that you all know very well, which is the Reynolds number. And what's the result of this? The result of this is, as George Whiteside so nicely showed many years ago in this cover of Science here, is the fact that we work within what we would typically call laminar flow regimes. So the idea here is that we're mixing a number of miscible fluids here, just varying the solute or the dye molecule in each. And you can see here that the flow and the fluid lamina are all moving here down towards the bottom left-hand side. And if we're going to try and mix these fluids, what we're going to have to utilize, in this case here at least, is going to be diffusion orthogonal to the flow. So this is very, very different to what we're used to on the large scale. And as I'm sure you know, it can give us advantages, and it can also make our life a little bit difficult. So laminar flow is typical. And then finally here's something which is going to be very important for the droplet work that I'll show you later today, is the idea that surface tension really dominates everything. And what that's going to mean for us is that we're going to be able to controllably form droplets or controlled emulsions in a way that's going to allow us to do experiments in a very fast and efficient manner. So this is all I wanted to introduce in terms of some of the reasons why we use microfluidics and some of the basic features of fluid flow. But I thought I would start off by just telling you a little bit of the work that we did a number of years ago now. So what I'd like to do over the course of the next few minutes is explain why we now adopt droplet-based microfluidic systems to do a wide variety of experiments. And the reasons for that are really encountered in some of the problems that we see when we work with continuous flow chemistry. But just looking back, I will talk about this piece of work a little bit later, but much of our early work in microfluidics was focused on performing both small and large molecule chemistries in batch and continuous flow reactors, and you can see a few examples here. We've just seen this. This is a very simple 3-layer device or 2-fluidic layer device, which allows us to mix fluids using the ideas of parallel lamination and allows us to access mixing times on the millisecond time scale. And once we were able to optimize this design, we could then apply it to important or useful and relevant small molecule chemistries. So you can see here an example, as you can see, many, many years ago now, but using this device here to start to make small molecule compound libraries. So the idea here is that by using this device, we can perform rapid mass transfer in terms of mixing of our reagents. In this case, we were looking at performing multi-component reactions. These are one-pot reactions which allow us to generate molecular diversity very, very quickly. But the problem with these reactions is that they're highly exothermic. So normally, to control the reaction, you need a huge amount of architecture around the reaction vessel. In this case, because we have a very high surface area to volume ratio, we can dump heat out very quickly, and the reaction becomes thermally flat. And that gives us control. So you can see here some simple examples where we could perform parallel, where we're varying the nature of one of our reactants here, or serial synthesis in a manner that we just couldn't do on the large scale. But after a few years looking and addressing some various aspects of small molecule chemistry, we became more interested in seeing if we could use microfluidics to start to control nanomaterial synthesis. So this here is a very short summary of work that we introduced back in 2001 here, but with a very, very simple idea. So as everyone in the audience here knows today, that nanoparticles and generally nanomaterials, not all nanomaterials, but most nanomaterials, have crystallite radii which are small relative to the Bohr radius. And what that means is that we can begin to think about tuning both the optical and the electronic properties of the material. The chemical composition, certainly for a lot of nanomaterials, remains constant. We just use the size to vary or to tune properties. And you can see here, if you like, the classic example here of a nanomaterial. This is just a cadmium selenide quantum dot. And the basic idea here is that the average emission wavelength here, the band edge emission, varies from the blue to the red simply because here we're making the particles small, and as the particle size increases, we can tune that band gap. Now, we're chemists, so what we like to do is make molecules, and in this case, make nanomaterials. So if we're using chemistry, how can we think about creating controlled or creating high-quality nanomaterials? Well, you can go back here to very, very old colloid theories. This is theory that's been well established and developed for colloids over the last 60, 70 years, but provides a very simple way about thinking about how nanoparticles form in bottom-up environments. So the idea here is that if we're going to make a nanomaterial, what we'd first of all like to do is we'd like to form a seed particle. So we can think about that very simply as a nucleation event. Then, once we form that seed particle, what we'd like to do is grow that to a particular size and then terminate that growth. So hopefully we're able to switch off things like aggregation, and we can normally do that very, very well. But the key challenge as far as the chemist is concerned is how do we separate nucleation and growth in space and time? This is the big problem that we have, because as soon as nucleation and growth overlap, what happens is that although we can control the average size of the particles that we're making very, very well, there's going to be a distribution in terms of the sizes within our population. And this can become very, very large when we're using batch techniques. So you can see here that although we require high monodispersity particles for many applications in biosensing, optoelectronics, etc., that's very, very difficult to do. So batch scale techniques are normally very complex. So they normally rely on trying to do the reactive process as well as we can, but then using some post hoc method to pull out the sizes that we're interested in. So it's a very long-winded way of saying chemists find it quite difficult to make nanomaterials, but if we can control nucleation and can control growth, then we should be in a much better situation. And we realized that a very simple way to do that was to ensure that mass transport and nucleation could occur very, very quickly. If we could do that, we could then ensure that growth and nucleation were separated, and that should give us a control over the particles that we made. So I'm not going to actually show you the original experiments to do that, but what we showed, and have since many other groups have showed, is that you can use continuous flow microfluidic systems to do exactly that. Because we can mix reagents rapidly, it means that we can control and discriminate those two processes. So once we worked out that microfluidic environments were actually quite good for doing this, we wanted to see how we could improve the situation. So what I would like to talk about here is some work that we began a number of years ago now, but still forms a large part of what we do, which is based around the intelligent synthesis of nanoparticles. So the idea here is that what we'd like to do is create a black box system to controllably synthesize nanoparticles that we define at the beginning of the experiment. And we're going to do that by combining a reactor, a detector, some kind of control algorithm, and then a means of changing some of the system variables. It's a very, very simple system. You can see it in this cartoon here. So we begin with a little microfluidic reactor here. This would normally, in our early experiments, be glass, single-layer device, maybe with a serpentine channel going through it. In more contemporary work, which I'll show you later, is simply capillary tubing. We have between 2 and 4 inputs, and we're going to rely on our microfluidic reactor to do the chemistry well. But importantly, once we've made our nanoparticles, they're going to exit the device, and then we're going to use this detection system to work out what we've made. So this is the big advantage of using microfluidics here, is our ability to integrate it with an optical detection system that gives us information. So if you look down at the bottom here, this is just a standard band-edge emission spectrum of a quantum dot. The important thing here is if I measure this, and I can measure this very quickly on a millisecond timescale, the peak here tells me immediately about the average size of the particles that we're looking at. The width of the band-edge emission here tells me about the size distribution. Any emission down here tells me about surface defects, and the intensity of this peak tells me obviously about the fluorescence quantum yield. So I get a lot of information that I can extract very, very quickly. And as you can see here, for example, by just varying this peak wavelength, we can then uncover or elucidate the radius. So the idea is I'm going to tell the system here to do an experiment. I'd like it to make a particular kind of nanoparticle. The detection system then works out what we've made. And then what we use is a control system which then judges how successful we've been in that experiment, and then it's going to change the variables of the experiment to repeat it, so hopefully we're going to get closer and closer to what we'd like to synthesize. And we make life very simple for ourselves by judging success just using this thing which we call a dissatisfaction coefficient. So this simply is a scalar, so it's a number between 0 and 1, and it tells us how well we've done the experiment. So here we've got some parameter gamma that can take a current value, a target value, and a worst value. And essentially, if we hit the target value, this collapses to 0. If we're doing as bad as you could possibly do, then it's going to go to 1. So we have a simple way to judge how well we've done the experiment. And in reality, what we would do is actually use a multi-attribute utility function here, where one parameter might be the peak wavelength, this one might be the intensity, and we can weight these according to our preference. So the idea is that we work out this dissatisfaction coefficient, and then we can either control temperature here or the flow rate. The flow rate controls concentration, and it also controls our reaction time. So if you look now at just some of the data, again, this is very old data here, but you can see how an experiment might go. So here we're varying temperature. This is the flow rate of our selenium precursor. This is the flow rate of the cadmium oxide precursor. Here we're trying to make cadmium selenide nanoparticles here, which are emitting at 530 nanometers. The wireframe cage here is our accessible parameter space, and every dot or point that you see there is one experiment. So the system has gone away, and effectively here it's done about 100 different experiments on its own, and the color of each of those points tells you how successful we've been. And you can see in this case here that perhaps unsurprisingly, because we want to get blue dots here, because these are our low dissatisfaction coefficients here, is that to make cadmium selenide nanoparticles which emit at 530, we're using generally low flow rates and high temperatures. But interestingly, you can also see that we're hitting good particles in regions which here are at low temperature or much higher temperatures that are very, very unexpected. This just shows you here some of the raw data, but this is the beauty. Because we can pull the information out so quickly, we have all this data which then allows us to vary things like temperature, residence time, reagent ratios to pull out information. So this kind of approach is very, very powerful in terms of pulling out information, and we use this to do lots of different things. More recently, and I won't spend any time on this, I should say that we've been applying metamodeling techniques, so increasing the sophistication of the way that we control our system. And this shows just a very simple idea where we're going to use, in this case, a Kriging metamodel here to try and predict what our hypersurfaces or our parameter space will look like for a real complex chemical reaction. So you can see maybe if we just look on the left-hand side here, first of all, how this might work. So this is something that you may have used before, certainly in MATLAB laboratories, the idea of using a Brannin test function here. So this simulates or shows us quite a complex hypersurface. So it's got a lot of peaks and troughs. And what we're going to try and do is use the Kriging, universal Kriging here to try and reconstruct that from a very small number of samples. So you can see here that what we've done over exactly the same parameter space here is we've randomly sampled, and I think it's 80 experiments here. You can see them by the black dots. And then from those measurements, we've then reconstructed the contour map that you can see up here. We then compare the actual map with the map that we've estimated here, and then we randomly sample another 200 points. And you can see here that we have almost perfect correspondence. So this is telling us that by using a metamodel, what we can do is start to predict very, very complex hypersurfaces using a minimal number of experiments. And just to give you some examples here, you can see data for the maximum, the full width half maximum, that's the width of our band edge emission, and the intensity. And if you like, if you just look at all these plots here, the basic idea is that how well do the dots correspond to the lines? And you can see that for most of these experiments, we've been able to really quite nicely model that parameter space. So the basic idea here is that we combine very nice detection and control techniques with microfluidics to do a good job, but we have a big problem. And that problem relates back to basic chemical engineering. The idea of pushing fluids through small pipes using hydrodynamic flows always gives us this problem here, and that problem is dispersion. So if we were to inject a beautiful delta function of material at the beginning of a reactor, as we push it through that reactor, because of our no-slip boundary conditions, we're going to generate this parabolic velocity distribution, and it's basically telling us that some material moves quickly, other material moves slowly through the device. Now that becomes particularly important if we're trying to control the growth of something. So ultimately, using a continuous flow to make nanomaterials is always going to be limited, and you can measure that using a whole different range of parameters. This is just our residence time distribution, and it essentially says that we're going to have very, very wide variations in our residence time. So unfortunately, we thought about trying to deal with this in many different ways, all of which were wrong. So initially, we started to think about how we can modify the surfaces. Can we make them super hydrophobic? That helped to some extent. Could we make them smaller? That again helps to some extent. But ultimately, it was far too long before we realized that what we needed to do was to start to move away from continuous flows and start thinking about using segmented flow. So this is really going to be the bulk of my, the rest of my talk, and I see time is already moving on quite quickly. And what we realized was that droplets can offer the chemist or the experimentalist a range of different possibilities. So you can see here just a couple of very simple videos and some pictures here. In fact, this is just an emulsion I made here with olive oil and food dye and water. And what you can see is that by using bulk shear forces, simply agitation in this case, what we're doing is we're creating a huge number of reactors. So in theory, at least, you can imagine that every single droplet you see here could be an isolated reactor which could house a reaction or a particular assay that we might want to look at. In this case, we've got water droplets which are surrounded by a continuous oil phase, and we can just control that through the volume fraction here. But we also have other problems. So the first problem, which is very, very obvious here, is that doing anything quantitative is going to be really, really difficult because we've got this really big variation in size. And then from the videos, the other feature of an emulsion, which we're all very aware of, especially when we're making something like a vinaigrette, is that droplets are unstable. So if we're going to use these systems to do quantitative biology, we really have to address each of these problems. And luckily for us, some really nice, beautiful work was done coming up to about 10 years ago now by people like Rustam Ismagilov at the University of Chicago, now down at Caltech, and a previous and recent speaker here at Waterloo, Dave Waite's group at Harvard, who showed how you could use very simple, again, passive microfluidic systems to start to make what I'm going to call controlled emulsion. So I'd like to show you some of the work that we've done, but again, I think it's really important that we just highlight here some of the key features of droplets or these controlled emulsions. So the first one here, and you'll see this in some videos in the next slide, is that droplets are completely isolated, and they're effectively going to define isolated reactors for which we can experiment in. We're going to be able to generate them using passive tools, and I've just shown two here. This is a T-junction, this is a flow focusing geometry. The basic idea here is very similar in each, is that we're just delivering two immiscible fluids. One of the fluids is going to wet the surface of our microfluidic device. The other one becomes the discrete phase, and the result of both hydrodynamic and surface forces is that we're either going to generate droplets here which are much smaller than the microfluidic dimension, or plugs or slugs that look like they touch the channel walls but are still separated by a thin layer of the continuous phase. So the first thing is that we're going to be able to make droplet volumes with precision. So we've immediately got rid of that problem we just saw, and I'll show you this in a video in a second, that our droplets are completely monodispersed, or as close to monodispersed as you're going to get. Secondly, as you push through, and I've marked these in blue because I think these are the really important reasons, at least why our group have started to use droplets to do lots of different things. So this one here is that mass transport now occurs without dispersions. Our droplets are going to move at a fixed velocity. That means we can time things. That means our kinetic measurements are better, and our control over reactive processes, by definition, becomes better. Secondly, we can vary the chemical payload of our droplets. I'll show you an example in a second, but essentially, for example, in this cartoon here, by varying the flow rates of these three input laminar flows, we can control what goes into each of these droplets. So we can create different experiments. And then finally, and probably most interestingly here, is this idea that we can make droplets very quickly. This is a very conservative figure. I would say that most experiments we work with focus on frequencies of around 1 to 5,000 droplets, or 1,000 to 5,000 droplets per second, but we can easily operate at flow rates, frequencies much, much higher than that. So the basic idea is we can make lots of droplets, we can control the size, what goes into them, and we can make them very, very quickly. So this, at least for the chemist and biologist, becomes very, very interesting. So you can see here, and I won't spend any time on these, but you can see here just examples, and I've seen some of these today in the labs here at Waterloo, of how we make droplets. So you can see in these examples here, using a standard flow focusing geometry, you can see the fact that we can make these beautifully monodispersed droplets. They're in different configurations here, they're of different sizes, but essentially by using microfluidic control, we exert control over the emulsions that we're making. So this is our first big bonus. Now secondly, as you can see in this middle one, again we're making droplets. What we're doing in this case is we're making droplets at a T-junction. I'm then splitting the detector channel into a red and green channel, because what I'm doing here is delivering a red fluorophore into our droplet through this laminar stream and a green fluorophore through this laminar stream. If you look at the red laminar stream, you can see that's got a constant width. We're keeping the flow rate constant, and that's why the droplets look uniformly red. But if you look at the green laminar flow, I'm actually varying the relative size or magnitude of that flow using a buffer stream, so that this laminar flow here is going to get thinner and thinner and thinner and thicker and thicker. And you can see that coming out here by a variation in the green fluorescence. So this simply shows that on a very, very fast time scale, we can controllably vary or dose what's going into each droplet. We can do other things. I'll come back to this later. This is just a device which allows us to thermally cycle droplets, change their temperature. You can do very simple things. Just by using a bifurcation here, we can split droplets and we can process them in different ways. We can take droplets that we've made and dosed in different amounts and isolate them and incubate them for extended periods of time. Now, this is particularly important for biology. We can synchronize droplets into particular orders, and then work here, which you would have heard about when Dave Waite spoke here at Waterloo, because this is a device that was developed by his group, and we've simply duplicated this for our experiments, is a device which allows us to sort droplets. Now, in our hands, we can't work at exceptionally high flow rates, but using a very simple detector here where we can look at the fluorescence of a droplet, and as it moves downstream, use an electric field to move it in an upwards or downwards direction, we can get this to work at between 2 or 3 kilohertz. So again, we can start to think about doing analytics on the droplets that we're making, but still on kilohertz frequencies. So before I show you some of the examples of work that we've been doing with these droplet systems, I just wanted to show you 2 other examples of passive systems that allow you to do very useful, very important unit operations. So if you look on the left-hand side here, you can see an example of a device which here allows us to merge droplets together. It's incredibly simple, but incredibly useful, particularly for synthetic reactions. All we're doing is taking a channel, dilating that channel, adding in 2 rows of pillars, and these pillars simply act as a filter for the continuous phase. So what happens, and you can see this if you went through or go through the images above, is that our droplet comes along, it effectively slows down and becomes pseudo-stationary here because the continuous phase is flowing around it. It waits for a secondary droplet, so it sits here, secondary droplet comes in, and by draining the continuous phase between the 2 droplets and destabilizing that interface, we then generate our merged droplet. You can see the internal mixing, and the droplet moves downstream. The lovely thing about this system is the timescale. It's about 30 milliseconds between a droplet appearing and the merged droplet leaving. So it's very, very fast. It's incredibly reproducible. And by varying the length and the asymmetry of those 2 pillar arrays, we can vary the number of droplets that we merge together. So this can be used to do quite complex chemistries. And then if you look at the middle video here and the images on the side, you can see actually a very similar device. This device allows us to do droplet dilution, something that we take for granted on the large scale, but something that's really, really difficult to do in microfluidics. And the basic idea here is that we introduce a mother droplet, which is very big for us. So this is about 5 nanoliters in size. We then deliver empty or buffer droplets, and through a process of merging, mixing and resplitting, what we can do is generate daughter droplets which move downstream. And if you've been looking, you'll hopefully have seen the optical density of the mother droplet and the daughter droplets is getting lighter and lighter as we progressively dilute. And the reason this works, again, is because this soft valve here provides a mechanism to initiate merging. And because we're using a continuous flow of our oil phase, we can remix the contents in the center. So the lovely thing about using a structure like this is that within, again, very, very short time periods, we can access enormous variations in concentration. So we can access about 4 orders of magnitude by going through about 20 of these merging operations. So this allows us to think about using the droplets in high throughput to do high throughput assays. So that's enough, I think, of the basic structure of some of the tools that we use. I just want to show you some of the work that we've done, again, over a number of years now. So the first thing that we actually wanted to use our droplet-based systems for was to start looking at single cells and doing experiments on small numbers of cells. And this is a paper we published back in 2007, but really is now the standard way that you would start to think about isolating small numbers of cells. And the basic idea here is that by selectively introducing here a buffer or growth medium with the cell suspension, you can control the number of cells that you release into this forming droplet, and then by using a detector at a particular point down the channel, you can probe some of the biology or chemistry that's going on. So you can see this in a slightly different configuration here, but it's just showing you the idea. If I deliver cells here, and these are very, very early experiments, so they're not very good, you can see the fact that we're making uniform droplets, and then whenever the cells come along, we can hopefully encapsulate these and then perform experiments on the individual cells. Now, something I won't go through today, but is a very important consequence of this, is that because the cells, or because the droplets can be made to be roughly the same size as the cells, dilution doesn't become a problem when you start to open up cells and do experiments. Even in microfluidic systems, the biggest thing that really kills you extracting information from single cell measurements is the fact that as soon as you open up a cell to look what's inside, things dilute very, very quickly. By ensuring that you encapsulate cells into droplets, you can ensure that that dilution is very, very small. And so what we showed is that you can do a whole range of experiments. You can see here, for example, where we're encapsulating single cells into droplets. By increasing the flow rate of the droplet stream here, you can see we encapsulate many more. And just to remind you, though, that because this is a random process, we're governed by Poissonian statistics, so this would be a limitation of the simplest embodiment of such measurements, is the fact that we've got control, as you can see here, of the average number of cells per droplet. But if we want to do purely single cell measurements in this kind of format, we typically have to operate, as you can see here, at conditions where most of our droplets, in this case about 90% of the droplets, are empty, and we maybe only have about 9% of the droplets, or 9.5%, contained in a single cell. So this is a limitation, but because we can operate the devices at such high speeds, our throughput remains high. Now, I'll come back to other ways which have been shown how you can get around this later on in the talk. And then this is a very quick example here, which shows you, I think, pragmatically at least, a really important aspect of droplet-based systems, and that's the fact that they're robust and you can integrate them with lots of different processes. So just speaking from experience, when you work with single phase flows, continuous flows, they're incredibly badly behaved, and that's predominantly because the molecules that you're working with see the surface. As soon as we move into a segmented flow regime, though, reproducibility and robustness really improves. So you can see here an example of quite a simple assay that we're doing here to screen photosensitizer activity for PDT, for photodynamic therapy. The basic idea is that we have a cell stream, we merge that with a photosensitizer. In this case, we're looking at small molecule photosensitizers. We can vary the concentration of these and the chemical identity. We make 140 picoliter droplets. We make these at around 650 hertz. We make about 500,000 of them, dump them into a reservoir. We can then expose the droplets in the cells within the reservoir to radiation for a given period of time. Then to see whether the photosensitizer has done its job and killed the cell, we can then deliver the droplets that we've processed, space them up a bit, add in viability assay agents, merge the droplets together, allow the chemistry to continue, and then screen them. And you can see here just examples of the raw data where these little rectangular features are the droplets, and these spikes here are the cells. If they're green and upward, that means the cell's still alive. If they're red and downward, the cells are dead. And then finally, you can build up these survival curves very, very easily. But the reason I'm really showing you this is that this can all be integrated in a very simple workflow. And this really comes down to the features that I've shown you about how these segmented flows can work in microfluidic systems. One example, and I'll go through this very, very briefly though, but this is something that I should have shown at the very beginning because this highlights one of the key problems, is that we used probably the simplest microfluidic device ever made, which was just a single channel going backwards and forwards many, many years ago to amplify DNA, but amplified in an incredibly fast way. So without going into details, the basic idea here is, if you look at that image, is that by resting a simple glass chip here on thermostatic copper blocks, we could reduce the thermal mass in the system to simply the volume of the fluid at a particular temperature. So this very simply meant for us that we could heat and cool reagents in a few milliseconds rather than a few minutes. And the result of this back in 1998 is that we could show here amplification of DNA in reaction times down to around 90 seconds. This is a very, very simple idea. So it was a very nice exemplar of why miniaturization can sometimes completely change the way that you can think about doing experiments. However, we had serious problems, and you can see these problems in this bottom bullet point here, and that's the fact that the high surface to area volume environment, which under normal conditions is so advantageous, in this case, first of all meant that our DNA, the template that we were looking to detect, and the polymerase enzyme was interacting with the surface, and that meant our efficiencies were very, very low. And then secondly, because we have a single phase flow, we had significant residence time distributions, which meant that timing was again a problem. And again, we worked on lots of ideas about how we could get through this. And again, it took us far too long to realize that the simplest way was to move from a continuous flow to a segmented flow. So you can see in this example here, we have a slightly different configuration. So this is a radial chip. So what we've done is made a chip which is round. We stick it onto a hot pin. So that means the center is hot, the periphery is cold. So to thermally cycle now, you just move your sample in and out like this. The only difference being that we've now segmented our reagents into droplets. So then if you look at a video here of the droplets being formed, you can see what's important. The idea now is our reagents are encapsulated within the droplets. They don't see the surface. And that means that in theory, our efficiency should be nice and high. And to cut a very long story short, when we look at the template concentrations that we're working with, if we're detecting signal from a template concentration of about 4 femtomolar, we know our droplet volume is 130 picoliters. That tells us that on average, I've got 0.3 templates per droplet. So on average, if I chose 3 droplets at the start of my experiment, I started off with 1 molecule of the template DNA in one of those 3 droplets. So that means we're doing single copy PCR. Our amplification factors are super high. And the real reason for this is very, very simply, we keep everything away from the surface. So this is clearly very important and has since, and before it, in fact, been used by people like Rain Dance to do some really beautiful experiments using droplets in a way I think that commercially hasn't been done before. So moving on very quickly, I wanted to go back again and tell you a little bit about nanomaterials. So I said that continuous flow systems were actually quite good for doing nanomaterial synthesis, but because the material interacted with the surface, that provided us with some limitations. What we can do, though, is we can use very simple microfluidics using the same ideas of flow, but now you can see we segment everything into droplets. So in this case here, as a simple example, we're making, in this case, for 6 semiconductor nanocrystals, in this case either lead sulfide or lead selenide, in PTFE tubing. We heat them by putting them through an oil bath, and then we do near-infrared fluorescence detection. So I just wanted to show you a couple of very simple features. So this figure here tells us that the obvious feature that by varying reaction times and by varying temperature, we control the average size. But if we look at the raw data, what we see is a lovely feature, and that's the fact that for these particles, by using microfluidics, we can make particles here which emit in this bluer region compared to the other nanomaterials. We're making here ultra small lead sulfide quantum dots, and these are things that we just cannot make on the large scale because we can't control nucleation. So this is a massive advantage. The other feature I wanted to show you here, again, just comes down to a pragmatic issue, is how robust are these systems? And you can see here that they are very robust. So this is just a snapshot, and I'm only showing you here data over a period of about 3 hours. We operate this system. This is the band edge emission of our product, and you can see that there's probably here around 1,000 curves that I've overlaid. They're showing exactly the same band edge emission, or pretty much the same. So this product that we're creating in this case is incredibly reproducible, something that wouldn't be the case in a continuous flow system, and you can evidence that both through the emission wavelength and also the band edge emission. So these environments, although they're very simple, are really allowing us to make very good quality material. So I know time is ticking on, so I just want to finish up with some more recent work that we've done before I then go on to talk briefly about some of the work we've been doing in detection. But this is recent work which we've been lucky enough to work with Maxim Kovalenko's group at ETH, where instead of using size-based tuning for nanomaterials, we're creating these perovskites, in this case cesium-lead halide and mixed halide systems, where we can tune the optical properties not by changing the size, or predominantly changing the size, but now by changing the chemical composition. So you can see here that by moving from the iodide through the bromide to the mixed iodide bromide, and then through to the chloride to the iodide here, we can vary the emission over a very wide range of the visible spectrum. So these materials are beautiful. The quantum yields very close to unity. The radiative lifetimes are up to about 30 nanoseconds. So these are hugely important materials. And we realized that by using, again, very simple microfluidics, we could start to do things that would be very, very difficult to do on the macro scale. So you can see here that what we're doing here is a very simple experiment. Again, we're using tubing. So we've moved away from planar chip formats. We have 2 T-junctions. This first one here allows us to vary our cesium to halide ratio, cesium to lead ratio. Here we're able to vary the halide ratio in terms of the synthesis. We then have a copper rod. So that copper rod has got grooves in it which we can wrap the tubing around. That first of all allows us to control the temperature really, really precisely, but actually more importantly means that we can, in addition to the fluorescence measurements, what we can do is we can pass a laser beam to do absorption measurements. So we can extract both absorption and fluorescence very, very quickly on a droplet by droplet basis. And you can see examples of that here. So we're making the perovskites online here. You can see the variation in their color. And maybe just to highlight a couple of features is the fact that we can screen conditions incredibly quickly. We can pull out the absorbance information here, something that we would find incredibly difficult to do in a batch scale system. And you can see here the time scales for the synthesis are super short here. Because we're controlling the nucleation and able to pull out consistent information, we're able to look at very, very early time reaction kinetics, something, again, that we just can't do on the large scale. My personal favorite in terms of this work here is the fact that we can use this intelligent synthesis again to make beautifully controlled nanomaterials. So you can see here, what we're doing is we're simply repeating the experiment, varying the experimental conditions, and making particles. And if you look at the emission characteristics, we can make them so that they hug very closely the extremities of this chromaticity diagram. If we use bulk techniques, we typically generate very low saturation materials, whereas here, these are highly saturated, and simply through that control of the flow, we're able to move them around to control both their saturation and their color. So I don't know how long I've got. Carolyn said 45 minutes. I know I'm already over 45 minutes, I'm guessing. So I'm not going to send you to sleep, I hope, in the last few minutes. I'm just going to very briefly go through something which I think is incredibly important, and I think something that we forget all the time. I'm not going to go through this, I'm not going to go through this figure in any detail, except if I were to go through it, it would tell you a very obvious fact. And that is, as soon as we get down to dimensional cross-sections below about 10 microns, working at normal concentrations, so here the concentration is a little bit low, but half a nanomolar, we have challenges in terms of looking at the molecules within our system. Life gets very difficult, and we need very good detection systems. And so we've spent a long, long time working out how we can improve that situation, or how we can develop techniques which ensure that we're pulling out enough information. So in the interest of time here, I'm not going to go through the details of this, but I'm going to just show you the results. One thing that we've worked with a lot is this technique of fluorescence lifetime imaging. Now, what this simply does is it moves fluorescence measurements from simply the measurement of a wavelength or of an intensity to the extraction of the lifetime, which tells you about lots of different parameters. And I'm just going to give you 3 examples here, and the examples of viscosity, concentration, and temperature. If you look on the far right, left-hand side here, this is one of the first videos that we measured using FLIM. What you're looking at is a channel where there's a dye at fixed concentration. The only thing that we're varying is the viscosity of the solvent. So the solvent here is water, the solvent here is glycerol water. So the flow is very uninteresting. Don't expect to see anything pretty here. But what we're doing is we're imaging the flow, and the color that you can see there is not fluorescence, it's a false color image of viscosity. We're able, in this case, to measure the viscosity of our fluid in or at video speed, which is incredible. What we can also do is, because in this case we're using 2-photon excitation, we can create 3-dimensional maps of the fluid velocity in a way, again, that you couldn't do using other methods. Now, in the center, this is concentration. This is what we've been doing more recently by using very simple hydrodynamic mixers, combining them with FLIM. What we can do is open up an understanding of how particular enzymes can repair DNA. And the reason we can do that is because of the extra contrast that the FLIM measurement gives us. And then perhaps my favorite here is by using FLIM to measure temperature. When we were developing the continuous flow PCR device I spoke about, I told you how it worked. That was very nice, but we had serious problems getting it to work perfectly. And one of those was because we had temperature heterogeneity within the system. So what we did here is an experiment where we're looking at a 50 micron by 50 micron chunk of a microchannel. We're heating it with a heater at the top, and then we're looking at the color here as a way of reporting temperature. So we're using a small molecule dye. Essentially, as it gets hotter, the lifetime goes down. As it gets cooler, the lifetime goes up. And that provides a beautiful proxy for temperature. And we can measure that with micron scale resolution. So this works really, really well. So for the last couple of minutes, I'm just going to show you in brief some of the results from very recent work that we've been doing to try and improve that situation. If you remember, right at the beginning of my talk, I said the reason we're doing what we're doing is to extract information. And actually what we do at the moment is very poor in terms of using all the information that microfluidics gives us. And I would argue that most of us here who use microfluidic systems are still very, very poor at extracting all the information that microfluidics provides. And you can see 2 examples of that here. This is work in our group. Again, using FLIM to work out mixing. This is work from Ismagilof's group back in 2003. If you look here, what he's doing is a lovely experiment. It's beautiful. He's using Droplet to look at some enzyme kinetics here. And if you look at maybe this image here, you can see that as the droplets move downstream, he's making a product that's fluorescing. The intensity of that gets more and more and more and more, and he can build up these beautiful kinetic curves. But look at what we're looking at here. This is all blurred, and that's because our droplets are moving too quickly. Now, if our droplets are identical, that's not a problem. because we can just repeat the experiment, and so he builds up these curves. But I showed you at the beginning, it's very easy for us to vary what goes into each droplet. In this case, if he varied the experimental conditions, he couldn't extract that information because we've got this blurring. So this is a big problem. And then again, in the same sense, for us to extract these lifetime mixing patterns here, we have to work on the basis that I can make many, 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 many identical droplets, and I can build that information up to generate our mixing patterns. So we began to think about, is there a very easy and direct way in which we can improve the situation? And as so many things go, we realized that technology existed for a very long time, which would enable us to do much more efficient extraction of information from droplets. And so what we're doing here is we're using ideas that were pioneered by Edgerton here at MIT back in the 1960s. The basic idea here is, as you know, is the fact that we can see this beautiful image of this bullet that's just gone through an apple is not because our shutter speed on the camera is very short, it's because we've been able to illuminate the scene with an even shorter pulse of light, or flash of light. So we're going to use exactly the same concept here. So for our droplet experiments, we're going to use CCDs or CMOS cameras which are incredibly sensitive, but have relatively slow shutter times, maybe here 10 milliseconds. But if we only illuminate the scene for 20 microseconds, we're going to be able to freeze the information and hopefully look at very fast processes. So this is an example, and I'm going to go through two very brief examples, or hopefully they'll be brief examples of how we've done that. So in the first one here, we're doing a simple kinetic analysis. We have a standard microfluidic device here, where what we're doing is we're mixing an enzyme with a substrate. In this case, we're doing a beta-gal assay, so we're just going to make, we're going to hydrolyze a non-fluorescent substrate to make a fluorescent substrate. And what we also do is we put in a barcode, and the barcode in our case is just a small molecule. So if we change the amount of substrate that we're putting in, we're going to also change the amount of the barcode, and we can use the barcode signal to back calculate concentration. So it's a very simple experiment. As soon as we encapsulate the substrate and the enzyme in droplets, they move through a few wiggles so we can mix everything up, and then through this serpentine section, and we're going to image the whole of this region here. Just for those of you who like optics, to remind you that we're going to use two lasers, one to excite the barcode, one to excite the product. We use an acousto-optic tunable filter to form our short pulse. We synchronize this with the shutter of our CMOS camera, and we're going to split the signal of the red and the green onto two halves of the detector. So that's how it works. I'm not going to go through the detail of the experiments except to highlight two important features. So if we look at this image here, this immediately shows you that using this stroboscopic detection has really helped us. This is the normal experiment with the continuous illumination. It looks like we've got a continuous flow here, but as soon as we strobe our excitation light, we can begin to see our droplets. And we can do that even if our droplets are moving at very high linear velocities. And then if you ignore this, because this is just some detail here on how we extract the information, but if you look at our kinetic analysis here, this is another beautiful feature of this system now. What we've done here is we formed a varying concentration profile of our substrate, which we report here through our barcode. And then all these blue lines, or the envelopes here, are giving us the kinetic information following the product. So if I was to look in a single droplet, at different times, I see my barcode signal is constant because that's reporting what we started with. The product is increasing, and from this data, I can immediately generate my Michaelis curve here. And what's so nice about this is that I can pull out my Km, which is spot on what we hoped it would be, and in this case, we've done 1,500 discrete experiments in 10 seconds. So we're not repeating anything, we're doing 1,500 discrete experiments. And you can see here that we've just used exactly the same microfluidic system, we've just been a bit smarter on how we've detected it. So I know my time is up, but if you bear with me for just 2 more slides, then we'll be done. And this is work that, again, we've been working on in different ways over the last year, and I think really now shows what you can really do in terms of high throughput measurements. So very, very quickly, What we're trying to do here is creating an ultra-high throughput imaging flow cytometer. So the idea here is simply in a normal flow cytometer, we would use a point detector to work out some characteristic of our cells based on forward, side scatter, or on fluorescence. What we're trying to do here is to get that information, but as well as doing that, being able to image the cells as they go through. And so what we're going to do is we're going to use something which I haven't had time to talk about, but something I'm sure many of you know a lot about, which is inertial focusing or inertial control of micron scale objects, in this case our cells within the system. We're going to use that to take a high concentration cell sample of around maybe between 20 and 40 million cells per mil, drive that through these channels so that the cells order, and then simply use this serpentine region where we have these channel switchbacks to then perform the stroboscopic illumination. So simple microfluidics, we're not using a sheath flow here at all, and an efficient detector. So I'm just going to show you the results of this, and hopefully you'll see why we're very happy about these initial experiments. So this again is just a cartoon of what we're doing. We have this flow manifold where we deliver the cells in, these 6 parallel channels where we balance both inertial lift forces and Dean forces to pack our cells axially and laterally, and then we have a little detector here. And if you were to look at a video, and this was in fact the very first one that we measured, these are examples of the cells going through the system that we're imaging. So we did some very simple measurements. You can see a measurement here where we're separating 10 micron sized particles from 15. That works very nicely. But we can also do cell imaging. So here we're looking at K562 and HL60 cells. The K562 is slightly larger here. We can extract the cell size from both bright field and fluorescence measurements. And here, by using our imaging data, we can do a 2-color experiment where we use a DRAC5 stain to highlight the nucleus and a wheat germ agglutinin stain to highlight the cell. And we can extract this information very quickly. So the 2 numbers, which I think are particularly exciting, is in this experiment, we're looking at a throughput of 96,000 cells per second. So not just the point measurement, but a relatively high resolution image of each individual cell. And you can see some of those here. So even at the high linear velocities that we're working with, which we have to work with, so these cells are moving at about half a meter per second, the blur is probably something less than 200 nanometers. So we get the throughput and we get the resolution of imaging. And we can use this to do lots of different things, which I won't bore you with, but this is an example here of using the technique to do cell cycle analysis, where we can discriminate between the G1 and G2 phases in our system. And you can see here data from a conventional cytometer really struggles to see this difference, whereas data from this system really allows you to do it. So I apologize for going over time. I remember that there was one more slide, but I'm not going to talk about this. This is simply to say that fluorescence is wonderful, but you need other detection techniques. So we've spent a lot of time recently working on vibrational spectroscopy, and also a technique which I'm very excited about, which is thermal lens microscopy, which allows us to look at non-fluorescent materials as well. So my final proper slide here is simply just to give you a take-home message, at least from my point of view, and that's, I think the microfluidic systems, as I've seen very clearly here in Waterloo today, are very, very good at performing chemical and biological experiments. But really to utilize their power and take full advantage of their power, we need very good detection systems to really pull out that information. And finally, and most importantly, I did very little of the work that you've seen today, so I want to thank all the people who did the work, who were up here, and I also want to thank the funders who allowed us to do the work. And finally, thanking you for your patience and attention. Thank you very much.
Thank you, Andrew. Very impressive, elegant way to control chemistry on the nanoscale and do these different ways of high throughput analysis. Questions for Andrew?
Chris? Well, I was going just in case nobody else might, but quick question, I hope. You covered a lot of ground that was very interesting, but I can't resist asking, How are you doing your Raman analysis?
How are we doing our?
Raman analysis.
From that second last slide there. So these are home-built systems. So it's not Raman. It's surface-enhanced Raman here for obvious reasons. So Raman is a technique, as you well know, Chris, which is analytically very desirable because it gives you, it's incredibly information-rich in terms of the information you can extract from it. The key problem in these systems is obviously the accessible limits of detection. So we work with both surface-enhanced Raman and surface-enhanced resonance Raman for particular applications. If I could have my way, Raman would be almost, or at least SIRS would be the best way for us to do many of our experiments. However, I should say that we can also do infrared measurements. So focal plane array detectors over the last few years have completely changed the ability for actually imaging with infrared to pull out information. And in fact, it's data I haven't shown here, but we published about 4 or 5 years ago that you can use infrared imaging to extract the same vibration information for segmented flows as well. So we effectively use, to actually answer your question, a confocal, home-built confocal technique, which allows us to do this, but it's going to be very substrate dependent. In fact, and just going back to that last slide, one of the, I know clearly won't go through this now, but some of the issues related to limits of detection have really made us think about other techniques. And this is a method which we've worked with Kitamori's group at Tokyo to develop photothermal spectroscopy. What's very nice about this is we can again look at non-fluorescent systems and effectively get a proxy for the absorption measurement, but at very, very low limits of detection. So I think this technique actually, in tandem with vibrational measurements, would be very, very useful for both small molecule and nanomaterial chemistries. So the bottom line is that you need lots of different detectors.
Very nice, thank you. There's an irony in that you're using SIRS after having spent so much time avoiding surfaces.
Yes, absolutely. So well, surfaces are really interesting. You know, that's true. I think the bottom line is we try and ignore surfaces as much as possible. And the surface does, you know, again, just from experience, the surface in microfluidics really controls everything that you do. Now, I've been very economical with the truth today in telling you that droplets are isolated. And droplets are isolated under certain conditions. And that means that at least on the timescale of the experiment, if everything's inside, that's a good thing. But again, the surface or the interface there is probably the most critical thing in droplets, and really understanding how we can control that. Again, I haven't had time to talk about it today, but we've spent a lot of time in the past 2 years trying to develop a better understanding of how surfactants work with our continuous and aqueous phases, and also working out how we can make surfactants robustly. And it's a very, very tough challenge. So that's still work in progress.
Thank you.
I have a few questions. At the beginning of the talk, you were talking about a control system for synthesizing. So can you elaborate on what was the control system for synthesis of quantum dots?
So the control system, well, so if you like, so the system simply started with the optical measurement. So by measuring the wavelength, the width of the band edge emission, and the intensity, that told us how close we were to a result. So in the first, if you want to know about the algorithms, I'm certainly not the best person to ask. So we were using SNOB-fit algorithms as a way of mapping parameter space. So the SNOB-fit algorithm effectively looks at the parameter space, and it basically mixes and matches local versus global information. So it doesn't focus on a local region where it finds a minimum or maximum and then sits there. It makes sure it really samples parameter space effectively. So we use that algorithm to then change the variables, so the reaction time, the concentration, and the temperature, and we then repeat that experiment again and again. So if I was to show you, and I don't have the data in this talk, but the dissatisfaction coefficient effectively drops down very quickly, and then we set some kind of threshold for the minimum drop per iteration. So you could continue this for a very, very long time, but normally within the tolerances you can do this quite quickly. So the optimization algorithms we use at a relatively low level, so the SNOB fit is. The metamodeling, I think, is much more powerful, because what that can do is in a very small number of experiments, it effectively then mimics or reconstructs a very complex parameter space. But, you know, we're not experts in this, so we've just really used the tools that we think are available. So it's a combination of all those, if that answers your question.
Spell the SNOB?
SNOB fit stands for Stable Noisy Optimization by Branch and Fit. But if you look on my website and the paper, which is Lab on a Chip 2007, the details of the algorithm are in that paper. It's a very nice algorithm.
Very nice work. Could you comment on how you recover your product after you've done the reaction in the droplet phase?
How do we
recover the product?
It depends what we do. So again, we're an academic group, so we're typically not making things for in large quantities, but essentially we can, in a lot of the systems that we use, say for doing the nanomaterial synthesis, you just break the emulsion afterwards. You separate the two phases and pull them out. Initiating the reaction is very, very simple, but terminating the reaction is clearly important for nanomaterials as well, because you either want to terminate the growth or stop aggregation. But you can use a microfluidic approach to do that as well. So our group, Jensen's group at MIT, have shown very elegantly how you can use little capillary splitters where you take the segmented flow and you can direct the continuous phase one way, take the product another way, and you can recycle it. So that's very, very easy to do. But you're quite right that being able to effectively recover your product is very, very important, because even though you're controlling what's going on in the reaction, there has to be a sharp stop at terminating things. But there are many techniques to do that.
Okay.
Very impressive. So, I'm interested in your way you do the thermal stuff with the droplets, and you get the header at the bottom here, and the droplet comes in, because the droplet moving so fast. By the heating, would that be sufficient enough?
Sorry, which?
When the droplet passes the heater, and you heat up the droplet to a certain temperature, is that what you do?
Was this for the PCR?
Yeah, the new PCR in the droplet phase.
Yeah, so
You get all the capillary wave dividing the
should be.
Yeah, when we're fast, there is a kind of connector.
We're gonna get there.
Yeah, this type of.
This one.
Yeah, the next one. Yeah, this one.
Oh, here. Sorry for the nanomaterial synthesis. Yeah.
Yeah, how high the temperature you heat it up?
These. I forget the exact figures, but we can go very high in temperature. The challenge there is to make sure that your continuous phase doesn't degrade. So you need to have very high boiling point. But we would typically be able to work between 300 and 400 degrees C.
When the droplet, because the droplet moving pretty fast, moving through the heating zone, the residence time, would the residence time allow the droplet to heat up very quickly?
Yeah.
So you're heating by what type of heating mechanism you're using?
In which case?
In this case, the heating zone, during the heating zone?
So the heating zone is just an oil bath at fixed temperature.
Okay.
So it's then just simply down to the fact that the tubing is in there. So again, it's the thermal mass of the fluid going in. But it is very, very fast. And often for these kinds of syntheses, you know, the reason that you heat for extended periods of time is because that certainly on a macro scale, you're not able to heat in a uniform way. But these systems work very well. I would say, however, that this is actually a better way to heat the droplets in this case.
Yeah, enough time for the heating up.
It gives them enough time. So I should also say that the volumes of these droplets are much bigger than the ones that I was talking with in terms of doing the cell measurements. But the ability here to both control the temperature, but also the ability to do that extra absorption measurement is really, really important here for us. So so we can, using this, it's to about 0.1 degrees C is the control we have.
Do you really measure the temperature of the droplet, or you just rely on the heat?
no. So the data, no, absolutely not. So the data, and I haven't shown it here, and again, the data is in the paper, is that we use FLIM to do that. So by doping in a small dye molecule into the discrete phase, we can monitor the temperature, and that's really, really powerful. But that's a very good question.
Okay, thank you.
Okay, in the interest of time, let's finish here, move the discussion to the back where food and drinks are waiting, and thank the speaker again.
Thank you.
Thank you.